What if scientists could take a piece of DNA from one organism and place it inside another? What if bacteria could be programmed to produce life-saving medicines? This is the reality made possible by recombinant DNA technology, one of the most important breakthroughs in modern biology. Over the past few decades, this technology has transformed medicine, biotechnology, and scientific research, opening the door to treatments that were once impossible. To understand its importance, we first need to understand DNA. DNA is the molecule that carries the genetic instructions for life. It determines how cells function and how the body produces proteins needed for survival. Inside every cell, DNA acts like a blueprint guiding the processes that keep organisms alive. For many years, scientists could study DNA but could not manipulate it. They understood how traits were inherited and how diseases affected the body, yet the ability to change genetic material remained beyond reach. Everything changed in the early 1970s. Researchers discovered special enzymes known as restriction enzymes. These enzymes act like molecular scissors, allowing scientists to cut DNA at precise locations. Once a DNA fragment is cut, another enzyme called DNA ligase can join the fragment with another piece of DNA, acting like molecular glue. This process, cutting DNA from one organism and inserting it into another, is known as recombinant DNA technology. The result is a new combination of genetic material that would not normally occur in nature. Although the idea seemed extraordinary at first, scientists quickly realized the enormous potential of this technology. One of the earliest and most important successes was the production of human insulin. Before the 1980s, insulin used to treat diabetes was extracted from animals such as pigs and cows. While it helped patients, animal insulin was not identical to human insulin and sometimes caused unwanted reactions. Scientists wondered whether bacteria could be used to produce human insulin instead. Using recombinant DNA technology, researchers inserted the human insulin gene into bacterial cells. Once inside the bacteria, the gene instructed them to produce insulin identical to that produced in the human body. This breakthrough changed medicine forever. For the first time, microorganisms could be genetically engineered to manufacture important medicines. Millions of diabetic patients now rely on recombinant insulin produced through this method. After the success of insulin, recombinant DNA technology quickly expanded into other areas of medicine. Scientists began producing therapeutic proteins such as growth hormones, clotting factors, and immune system proteins using genetically modified cells. These treatments have improved the lives of patients with hormone disorders, blood diseases, and immune deficiencies. Vaccines also benefited from recombinant DNA technology. Instead of using weakened viruses, scientists could now produce specific viral proteins in laboratories. These proteins stimulate the immune system without exposing patients to the entire virus, making vaccines safer and more precise. Another exciting development made possible by recombinant DNA technology is gene therapy. Many diseases are caused by faulty genes. Gene therapy aims to correct these problems by introducing healthy genes into a patient's cells. Using recombinant DNA methods, scientists can design genetic material that replaces or repairs defective genes. Although still developing, gene therapy has already shown promise in treating certain inherited diseases and rare genetic conditions. Beyond medicine, recombinant DNA technology has also influenced agriculture. Scientists have developed crops that are resistant to pests, diseases, and environmental stress. These genetically engineered crops can increase food production and reduce the need for chemical pesticides. However, the ability to manipulate DNA also raised ethical and safety concerns. When recombinant DNA technology first appeared, scientists debated whether genetic engineering might pose risks to society or the environment. These discussions led to the creation of strict guidelines and safety regulations that continue to guide biotechnology research today. Despite these concerns, the benefits of recombinant DNA technology have been remarkable. Many modern medicines used to treat cancer, autoimmune diseases, and rare disorders are produced using recombinant DNA techniques. Biotechnology companies rely heavily on these methods to develop innovative therapies and medical products. This technology also paved the way for newer tools such as gene editing, including advanced techniques that allow scientists to modify genes with unprecedented precision. In many ways, recombinant DNA technology marked the beginning of a new era in biology, an era in which humans could not only study life but also modify it to improve health and treat disease.